So thank you all for your patience, incredible patience. And thank you for coming and braving being indoors with us and for coming out on a cloudy Wednesday. We are the Bay Area Lyme Foundation, and we're a nonprofit foundation started in 2012 in Silicon Valley by a group of women who were very concerned about some of their own and some of their friends' misdiagnoses before getting properly diagnosed and treated for tick-borne diseases. Important to some of you who might be PhDs or researchers, medical providers, we basically give grants to scientific researchers across the country. We also do other things that I'll tell you about, but primarily you can hold on to the concept that we are a granting organization. We have awarded over 100 project grants across the US at 37 institutions. And we started something that's been kind of fun over the past few years is a citizen science project where people can send in ticks from across the country, and they were identified and tested and put onto interactive maps that came out recently. You might have seen them in the news. And they showed ticks and tick-borne diseases in places where the CDC and state governments had not previously detected them. Our education program, which I'll talk a little bit more about in a couple minutes, has reached over 2 million children. I know one of you earlier at the table was telling me that you feel like every single child needs an education on tick-borne diseases. It was you, Dr. Haney. And we agree. And so our goal is to try to get as many youth educated. We're starting with the state of California and moving outwards from there. We have established a new national Lyme disease biobank. The Lyme disease biobank PI is here today, Dr. Liz Horn, and she'll be speaking to you about that. And over the past 9 years, we've raised more than $36 million to fund novel research for the prevention, diagnosis, and treatment of Lyme and tick-borne diseases. And that is kind of amazing. Yeah, considering we're one of the biggest funders of Lyme disease research in the country and the biggest nonprofit funder of Lyme disease research in this country. We've sponsored 7 influential Lyme conferences, including the Stanford, Harvard, the first West Coast Stanford, Harvard Tick-borne Disease Conference a couple of years ago. And I know some of you were there. And we've hosted 21, maybe now 22, speaker series across the US from California to New York. Some of our research collaborators, of which there are many, we work with MIT, Stanford, Harvard, Tulane, Duke, Penn, the Broad Institute, UC Irvine, UC Davis. We are very excited to have new researchers on board. So if you know anyone who would like to submit a project proposal, they're sort of NIH-style grant applications, and we have rolling application acceptances. So just quickly, you can see that we've grown in our resources and our funding, through the past few years. And we divide our research funds into mostly, our tagline is, we aim to make Lyme disease easy to diagnose and simple to cure. And so you can sort of see that in this pie chart where we allocate a certain percent to diagnostics there, 39%, and it was 36% on Lyme therapeutics in the year that in 2020. We also work on prevention, which is the education piece. And I'm the director of, I should introduce myself. My name is Leah Gertner. I'm the director of education and outreach at the Bay Area Lyme Foundation. So it's a smaller part of what we do, but I know a lot of you who I spoke to earlier feel like it's a really important and critical piece of what we do. And Lyme ecology is another portion of where our research funds go. And so you might have seen after the COVID vaccines came out this summer, and there was that little blip where COVID wasn't on the front page every single day. There was room for our latest study, which was our tick ecologists found Lyme and tick-borne diseases in ticks in the California coastal areas near the beaches. And so the grasses near the beaches were in Marin County, where I'm from, up to 30% of the ticks that they tested carried a tick-borne pathogen. And so that was sort of startling information, and it made it into the Washington Post and NPR. And that's just a small study that we funded, but that's the kind of thing that people hear about, and it brings attention to the disease, and so we do allocate some of our resources that way. Prevention and education, as I mentioned, is an important part, and I'm bringing it here because this is something that you can access on our website. You can download tick education, tick bite, prevention, educational material on our website. You can watch videos. You can see our previous speaker series on our website. I have a Tick-tective podcast and video series where I interview researchers and patients and people who are prominent in the tick-borne disease world. So you can learn anything from researchers at the Broad Institute who worked on COVID and Lyme sequencing, all the way to the first female jet fighter pilot who's in the National Hall of Fame, who was medically retired due to a tick bite and is an active speaker on that topic. So if you have any questions, please come up to us later after the talk, and I would like to move on and introduce Dr. Liz Horn, the PI of the Lyme Disease Biobank. Dr. Horn has a doctorate in molecular pharmacology and cancer therapeutics from SUNY. She has a master's in business informatics from Oregon Health and Science University, and she is a National Library of Medicine Fellow in biomedical informatics. Go over, Dr. Liz Horn.
Great, thank you. Oh. Okay, so I just wanted to check. I was going to take my mask off to give my talk. Are you guys all cool with that? Okay. No, it's important to ask. We want people to be comfortable. Okay. And there is no clock, so I'm going to set my timer. So I'm a little out of practice, like many of us, because we used to do this stuff all the time and then we didn't. And so yeah, it's good I'm setting a clock. Anyways, so thank you so much. I wanted to tell you a little bit today about what we're doing with the biobank, and we're starting an initiative in San Diego. So I'm going to wrap up with that, but really I want to give you a little bit of information of where we are, what we've done, and where we're going. But I wanted to kind of start to get us all on the same page about what is Lyme disease. So we all know it's a bacterial infection. It's caused by the bacteria Borrelia burgdorferi. It's transmitted to humans by an infected black-legged tick bite. And what's interesting is the ticks are different on the East Coast and the West Coast. So on the East Coast, it's usually Ixodes scapularis, the black-legged deer tick, and on the West Coast, it's Ixodes pacificus. So it was identified almost 50 years ago in Lyme, Connecticut, and in acute stages, you get a viral-like illness. And it's common in endemic areas, and we really think about an endemic area like the East Coast and the upper Midwest, but there are parts of California that are endemic, and especially with the new study that the Bay Area Lyme commissioned with their tick ecologists, there are ticks in the grasses in coastal areas, which I didn't even think about that. I know about tall grasses and be careful about them, but I think one thing about ecology is if you want to know if ticks are there, you need to look for them, and if you don't look for them, you won't find them. And in California, tick season is year-round, and also in places where winters are mild, so that makes it even more challenging because it can compete with cold and flu season, and now we have this new thing called COVID. Ticks are really tiny too. And if you think about an adult tick that's on the left, that's about the size of a sesame seed. And on the right is the nymphal tick, and that's the size of a poppy seed. So they're very, very tiny. And in early Lyme disease, which is where I spend most of my time thinking about this, and I think this is really important in California because it's not as common, and I think for medical providers, they're not really thinking about Lyme in California, but we know it's here. And so we often get an expanding Lyme rash or a lesion, and it's also called an erythema migrans, so you might see it referred to as an EM, and it usually occurs within a month. It's round, oval, or regular shaped, and it may or may not have central clearing. And that's really important because you hear a lot about, try to be fancy and see if there's a pointer. Oh, pointer. So this is what someone would call a classic bullseye rash, but it's not very common. And I think that if you're a provider or even a person walking in the woods and you get bit by a tick and you're looking for this, and you get this, you might not identify it. So this is what we actually see more commonly in our study. Homogeneous erythema, you can see where the tick bite is. This is actually called a punctate or a punctum, and you can see the skin kind of peeling around it. And there are a lot of studies about how often Lyme rashes occur, depending on the study, 43 to 70%, but it's not all the time, and I think that's really important. And rashes can also be difficult to see because they can be in places like your scalp or on your legs or places with hair. So I think it's important to be aware of them, but we don't always get them. And I just have a variety of some of the different skin rashes in our study, and you can see some are really red. On people who have darker skin, they're often harder to see. They can be more challenging in folds. So this is kind of in like the armpit area. And they really look different. And sometimes they're very big and sometimes they're smaller. So I think it's important for especially providers to realize these can look different. And then we also have tick bite reactions, which they're not technically not EMs. That's sort of potentially the beginning. Not all ticks carry the bacteria that causes Lyme disease. So if you see this, this isn't an EM yet, and we don't know in these cases if it will progress to one or not. In terms of early Lyme, within days of exposure, you get flu-like symptoms, headache, fever, chills, fatigue, muscle aches, and it's hard because it overlaps with other common viral illnesses, except you typically don't get respiratory symptoms or cough. And then within weeks to months of exposure, and this is if you're not treated, you can get musculoskeletal pains, arthritis in one or more joints, nervous system problems, and heart problems. While they're rare, occurring in less than 10% of people, they can be pretty severe. And within months to years of exposure, you can get a lot more complicated medical issues and with neurologic complaints, neurocognitive effects, brain fog, and profound fatigue. So I briefly touched on some of the different stages of Lyme disease, but I wanted to just point out that there is this fantastic CME website for physicians, and we need to get the word out. This is run by Dr. Elizabeth Maloney, who's a retired family practice physician from Minnesota. And she's done some really nice modules. And I just took a screenshot of the homepage, and these are all free. But this is when a black-legged tick bite is more than a bite. So how do you differentiate if it's a bite or if it's actually early Lyme and you need treatment? And we did one with her for managing Ixodes pacificus bites, because these might be different than managing eye scab bites. And then she also has fantastic things on diagnostic testing and serology, because diagnostics are complicated in Lyme. And we have challenges, and one of the biggest challenges is unreliable diagnostics. And that's really why we created the biobank, to get samples into researchers and companies' hands to be able to create better tools and diagnostics for Lyme disease. And Lyme disease is often misunderstood, especially in California. There's different species and strains than on the East Coast. And you hear people say things like, oh, there's no Lyme in California. Well, that's not true. And so Another reason we created the biobank is to build the body of evidence and to collect samples because we can change people's minds through data. And one of the huge challenges, this is not just in Lyme, but the slow pace of research, limited federal funding, and I think especially now there's been such an emphasis on COVID, rightly so, but we all need to get back to studying all of these other diseases that are important, including Lyme disease. So I'm going to tell you a little bit about the Lyme Disease Biobank. This is Bay Area Lyme Foundation's biorepository. There's really three arms to it. There's all the things we've done in early Lyme disease, which is where we started, and I'm just going to touch on that. And then I'm going to talk about what we're doing in California, and then I'm going to talk about our tissue initiatives. So we started collecting in 2014 with a pilot study at a site on Long Island in East Hampton. And now we have over 1,000 participants that are enrolled, and we have early stages of Lyme and other tick-borne infections. And very often, I'm just going to say Lyme disease, but please know I mean Lyme and other tick-borne infections because we know Lyme doesn't always travel alone. So just add another TBIs when I say Lyme disease. We also collect healthy endemic controls, which is really important because these are people that may be exposed in their everyday lives because they live in endemic areas, and that's a really important comparison. For some of our samples, we collect ill controls. So we collect samples from people who have been hospitalized with tick-borne infections. And so to have a good control sample, you really need someone else who is ill with fever that's in the hospital with something that's not tick-borne. We have carditis samples. We're collecting neuroborreliosis samples with appropriate neurologic controls, as well as persistent and chronic Lyme. And we've really set this up so each sample donation can support approximately 50 research projects. We have more than 65 projects in academia and industry that are using samples, and we've distributed more than 12,000 aliquots to researchers. So aliquot is just really a small piece of the sample. So you get like a big blood tube, and then you aliquot into smaller bits. So think about like if you're cooking something big like soup and you want to freeze it, you don't freeze all the soup together, you put it in kind of smaller containers. So we've distributed 12,000 of these to researchers. These are our sites, and I'm really excited about the work we're doing in California, which I'll tell you about a little later, but we're collecting on East Coast, on Long Island, in Boston, in the upper Midwest. And I think these are areas where we are known to be endemic. So it's important to have samples from there, but also samples from California. We're working with an urgent care facility in the Bay Area. So our early Lyme disease sites are in red. Our later presentations and kind of that sort of sicker patient that's going to be hospitalized, that's in light blue. And our sites for persistent and chronic Lyme, we're in the San Rafael area, as well as in San Diego. And I'm going to tell you a little bit about our work in San Diego. So, I'm going to just close your eyes really quick. So we published a study in last May, in May of 2020, and we characterized 550 samples. And so what was cool about the study is we enrolled people based on signs and symptoms of early Lyme. So we enrolled people with EMs and people without. And that's important because not everyone presents with a rash. And when they were coming to their doctor to be evaluated for Lyme disease, we asked them if they wanted to enroll in the study. And so what you're seeing here is these type 1s. Oh, sorry, that was not the type 1s. They have rashes, the type 2s don't, and these are endemic controls. The Cliff Notes version is down here. But we've enrolled more men than women as cases, and we don't really know why that is, and it's across all the sites, because you would think men and women would get exposed easily in an endemic area, and this is something we want to do more research on. The majority of this is the mean age. We sort of have an older cohort, and our cohort, the majority of it is white, but at our East Hampton site, we have about 20% that are Latinx, and that's because we have Spanish consents and bilingual staff at our East Hampton site. And that's really important because you want to see Lyme in different people. You want to see it across in men and women, in different races, in different ethnicities. So we're trying. And so the key findings is we collected these 300 samples, And then we test all of our samples for Lyme disease, right? And Lyme uses a 2-tier testing algorithm where you test with an ELISA on the first tier, and if it's positive, you do the Western blot. But we said, no, we're not reflexive testing. We're testing everyone. We're even testing the controls because we want to know what's the background. So we actually have all of that data, and it's super valuable. So controls without history of Lyme or tick-borne infections, they don't test positive for Lyme disease, which we would expect. But what we were really surprised at is only 27% of our cases were laboratory confirmed. And we all talk about how the diagnostics aren't meeting the needs that we have, and our data really supports that. This talks about all the different groups, but I'm just going to skip over that we enrolled because we stratified our cohort into different areas. And the people with clinical diagnosis, so they had an EM greater than 5 centimeters, those that were 2-tier positive, it was only 29%. So that's a real discrepancy. And I'm going to show you some cases just to give you an idea of why this is a huge issue. And we're trying to understand this more with some additional testing. And then the other thing that's important to this is there's a thing called seroconversion, where your immune system changes over time in response to a pathogen. And so you have different antibodies. And in the beginning, you look at IgM, and later on, you look at IgG. And IgG is supposed to go up, and we don't see it going up. And we see it in less than 5% of the people that were laboratory confirmed. So we're doing more work on this. But the main point of this is it highlights and reinforces our known limitations of serologic testing in early Lyme disease. So we also know there's problems with our tests in persistent and chronic Lyme, but we're also seeing it in early Lyme. So close your eyes really quick because I don't want to make anyone nauseous. I just want to show you 4 quick cases. And I showed you rashes before that didn't have central clearing, but now I'm going to show you ones that, like, these are classic Lyme rashes that a clinician would just say, yes, this is Lyme disease. So this is our first case. This is a 43-year-old male. He doesn't remember having a tick bite, but they're small, right? We're talking sesame seed, poppy seeds. It's okay, he doesn't remember. He's had a rash for 2 days. He's had fever, chills, headache, and dizziness. No previous history of Lyme or other tick-borne infections. And in clinical care, when you present with this, they don't order blood tests because they're like, okay, you have Lyme disease. But we test everybody. So this is someone that would be a clinically confirmed case of Lyme disease by a provider in an endemic area. And this is just information about where we did the testing because we tested in different places. These are all CLIA certified labs. And they have negative PCR of blood, and it's really hard to find spirochetes in blood. And their first ELISA was positive, but the Western blot was negative. Wait, I'm sorry. Sorry, this guy's 2-tier positive. So the ELISA is positive and the Western blot is positive. So this person is laboratory confirmed by standard 2-tier testing, or STTTA, which stands for standard 2-tier testing algorithm. And we got the same results on the second draw. The IgG never came up. So this person's immune system behaved kind of as we would expect it, except for the IgG. So this second person, this is another guy, 31 years old. He reports a tick bite 3 days ago. You saw this person earlier with a nice central clearing. Like, this is a beautiful, beautiful EM rash. Has a bunch of other symptoms: fever, chills, fatigue, headache, night sweats, nausea, on and on. No previous history of Lyme or tick-borne infections. Again, don't run tests in clinical care because this is Lyme disease. This person was enrolled in Wisconsin. Laboratory testing was the Mayo Clinic. They were positive by PCR, and that's pretty unusual in Lyme disease because the spirochetes do not hang out in the blood, they hang out in the tissue. So we'll talk about tissue at the very end. And this person's negative on all serologic tests. Lyme disease, and here's a woman, this is her arm, and you can see some nice central clearing here. 40-year-old female, doesn't recall a tick bite. Again, not uncommon, ticks are small. She's had a rash for 3 days. She doesn't have any other symptoms other than her rash. No previous history of Lyme or tick-borne infection. Again, lab tests not ordered because this is Lyme, gets prescribed antibiotics. And she's negative on all serologic tests, and we do a second draw on her 2 to 3 months later, and she's still negative. So she's not behaving the way, her body is not behaving the way we would expect. Her immune system is not behaving the way we would expect and what we would expect to see with testing. And this last one, this is kind of a fun, so this sweet guy is a tattoo here, so that's why he's covered up. He's 53 years old. He doesn't recall a tick bite, has a rash for 14 days, fatigue and headache, negative PCR, positive on the first tier ELISA, and negative on all serologic tests. So out of these 4 cases I showed you, only the first one would have been 2-tier positive, even though they all have really nice, well-characterized Lyme rashes. So we have a problem with diagnostics in early disease. Generally, we need better Lyme diagnostics. And I told you all about this, so we're not going to tell you again. And I want to let you know that even though we started in early disease, that wasn't our complete goal, because we know chronic Lyme is a huge, huge issue. It has huge burden, and that's an area where we really wanted to get more involved with. And we also wanted to get involved in California because we're based in California. I technically live in Oregon, but the Bay Area Lyme Foundation is in Portola Valley. And Lyme disease is the fastest growing vector-borne disease. This number's been updated by the CDC. It's not just an East Coast problem. They've identified infected ticks in 42 of 58 counties. This is from the California Department of Health, but they don't test in all the counties. So if you don't look, you won't find them. And we talked about the symptoms. They can be nonspecific. If it's untreated, it can be really debilitating. And so we need samples from infected individuals in California. So we've been working on a project in San Diego, which we actually started in the end of 2019, and we had a great ribbon cutting ceremony in March of 2020. And that's Tika over there, our research coordinator, that will be enrolling people in our study. We have almost 20 samples from San Diego. We have these great little pamphlets out there. If you're a provider, or if you just want more pamphlets to give to other people, we have little bags with them. We're happy to share them with you. And on the back is a QR code that tells you all about the study and what's expected. We compensate participants with a $50 Amazon gift card as a thank you. It's 1 blood draw, and there are some questions to answer about your medical history. So if you're interested, and we're giving these samples out to researchers, we probably have about 5 groups studying the persistent sample cohort we have so far. We have about 75 samples, and we're getting some really interesting results. And again, this is Lyme and other tick-borne infections. So this is an example where you have to add that on. And just really quickly, I want to tell you about the tissue bank. My background's cancer research, so I really think if you want to see what's happening in the body, you need to look in the tissue. And so we have been collecting samples from people with Lyme disease, with chronic Lyme, that are having surgery. Our focus has been knee and hip replacements, but we do others on a case-by-case basis. We work with the hospitals. We have a partner, NDRI, that helps us do this work. We also partner with My Lyme Data, and NDRI walks everyone through the process. And I would say the only thing about this is, if you are having surgery and you are interested in providing a sample, is we need a little bit of lead time, just because every hospital has different rules and we have to work through their system. So we need at least a week. So things that are emerging surgeries that, like, I hope this doesn't happen to you, but you have to get your appendix out tomorrow. We're not able to be nimble in that situation, but we are really excited about getting these samples to researchers, because we want to look for spirochetes in the tissue and see how spirochetes change what's happening in that tissue microenvironment. And I want to just leave up my acknowledgement slides. I could not do this alone. We have a lot of different partners. We're incredibly grateful. And I just want to briefly tell you about the other things we have on our table. We have a flyer about the tissue bank, and it talks about the process and who to call and what to do, and feel free to take any of these. And then we also have some fast facts about Lyme in California. Yes, I use a duck. I think it's funny. And the dog loves it. The dog's like, what is that? Anyway, so fast facts about Lyme in California, because it's really important that people understand there is Lyme in California, and it could be different than what's on the East Coast. And this is just what we're doing in San Diego. And here's a picture of our ribbon cutting. And I just want to acknowledge Sharon Wampler, who's sitting in the first row, who really made that happen and got Representative Scott Peters here. And He's the tall guy in the picture, and other representatives from San Diego government, and really getting people to understand in San Diego that Lyme is a big deal and it deserves our collective attention. And thank you, and I think I went over by 30 seconds, but like I said, I'm out of practice.
I'm going to ask that you hold your questions until we have a panel, if you don't mind waiting. I hope you all make it to the end. Yeah. So I would like to introduce Dr. Victoria Blaho. She's an assistant professor in the Immunity and Pathogenesis program of the Infectious and Inflammatory Diseases Center here at Sanford Burnham. And she has a joint appointment in the NCI Cancer Center. She was awarded the first ever Obede Award for her work in sphingolipids and has studied the immune response in Lyme disease, multiple sclerosis, and leukemia, amongst other things. She did a fellowship at the Weill Cornell Medical Center and was a Leon Levy Neuroscience Fellow. She was also awarded a Scientist Development Grant by the American Heart Association, and her BS in biochemistry and her doctorate are in molecular microbiology and immunology from the University of Missouri, more or less, right? Okay. Okay, thank you for coming to speak, Dr. Blaho.
I'm used to seeing my computer have the timer. I'm going to use that. So. I can get very loud too, so please tell me if I start to amplify my voice too much. If I get too excited about something, it might get a little too loud. All right, well, thank you for such a nice introduction. And good evening now, and welcome to Sanford Burnham Prebys. I have to say it was a real honor to be invited to speak to this group today, and I'm very excited about the opportunity to share my love of infectious disease immunology and the science behind Lyme disease. So today I'll just start out by giving you some overview on the immune system and how immunity develops, and then tell you about how the immune system uses that immunity to confront the invasion of pathogenic organisms, especially in this case, Borrelia burgdorferi or various Borrelia species. And then, since we got such a great overview of Lyme disease from the earlier two folks, we won't really have to go over too much Lyme disease background, luckily. And then I'll share with you some of the previous work I've done over the years looking at the role of a very powerful biological signaling molecules called eicosanoids or lipids, and how they may be regulating the development and the resolution in a mouse model of Lyme arthritis. And now with the biobank, it's a really exciting possibility of maybe getting human samples to test out some of our hypotheses that we've had for a really long time and haven't had any opportunity to really test with human tissues. So quick notes for anybody who's in the know. If I say Borrelia burgdorferi, I mean that in the sensu stricto sense. Anything that is the sensu lato, I'll use the species name, but I don't think I really go over any of that. So because of COVID, I'm actually going to be using a, let's see. Oh, what's that? Pointer. Yeah, I know. This was, well, my lab assistant is in here and he would tell you that he's not surprised that there's something going wrong with the computer right now. So, yeah. I did. I warned you. I warned you. I follow. Oh, hey. There we go. All right. So, thanks to COVID, I'm going to make a statement that I never would have anticipated saying before COVID, which is most of you are probably familiar with some aspects of the immune response. Never ever said that before in my entire life. Very weird. But the immune system is pretty complex, and there's probably a lot of questions that you have about how all of these different parts might fit together. So there's a lot of different components in a very basic sense. Aha. In a very basic sense, you have your immune cells. You start out with your innate immune cells, the neutrophils and macrophages. These are the ancient immediate responders. They show up right away. Tissue damage, infection, they're the ones that are there. They're followed by T and B cells, which are the acquired immune cells. They show up later. You're familiar with B cells in a way because they're the cells that make antibodies. So they secrete those, and then they go into circulation, and they are considered soluble mediators of the immune response, along with cytokines and chemokines. You're probably also familiar with a certain cytokine called interferon. That's just one of many, many cytokines and chemokines that are responsible for regulating different signals between different cell types to tell them what to do and where to go. And then also, we have my favorite soluble mediator of immunity, lipids. Lipids can be very simple. They have a simple structure with just a few carbons and some phosphates and some oxygens on them, or they can be very complex, like here you have high density lipoprotein or HDL, the good cholesterol. So all of these work together, going through the body, through our circulation in both lymphatic vessels and in the blood vasculature. And they are generated in bone marrow, thymus, and then they go through spleen and different lymph nodes to travel through the body into different tissues to surveil and to assess for danger. So the whole point of this very elaborate system is that the organism is trying to survive. Whatever that organism is, it is trying to survive through immunity. Immunity, or the generation of immunity, is the result of an ancient struggle for survival. You can start out with man versus wolf, and as you travel back through bison eating grass that have a fungus on it, you have fungus that are infected by bacteria, and you have bacteria that are infected by viruses. And all of these have developed specific and special immune responses to combat this for the purpose of identifying danger, correcting damage, and surviving. The organism must survive. And as humans, we spend a lot of time anthropomorphizing things and saying, this thing is trying to get me, this bacteria is trying to get me, this dog is trying to, well, the dog might be trying to get you, but the virus is trying to get you. But none of these things is really trying to get you. It's just trying to survive, and you're an unhappy, unfortunate victim of that instinct and that need to survive. So again, for instance, you have this bacteria, which is a streptomyces, and then all of these little things hanging off of it are phages, which are viruses. And the reason why I thought this was kind of interesting is that some of you might have heard of a method of gene modulation called CRISPR-Cas, and this was awarded the 2020 Nobel Prize in Chemistry. This is how streptomyces, or one of the ways that streptomyces has an immune system. It has CRISPR-Cas, and this allows it to kill phage viruses. So even all the way down to bacteria, there are numerous methods in place to institute an immune system. And so we have to keep that in mind when we try to figure out how to prevent or how to cure or how to eradicate an infectious disease. Because we're not just dealing with our system and our desire to get rid of it, we are dealing with its desire to survive. And so some of the things that we don't really think about a lot of times, however, the Lyme disease field is probably one of the few fields that does actually take this into consideration, is whether or not that infectious organism has a reservoir. Does it have vectors that transmit it? Does it have different physiological states? Does it have a cystic form? Does it have a budding form? Does it have a sporulating form? Diseases that have only a human reservoir, they're not found anywhere else on Earth, as far as we know, that are passed only human to human and have a single state as an infectious agent. It only exists as one thing. For example, smallpox and polio are our best chances of getting rid of something, which is what we did with smallpox and would be able to do with polio. However, that is definitely not the case with Lyme disease. So there are more and more studies coming out showing how many, very many reservoirs there are for Borrelia burgdorferi, for Borrelia species out in our environment. Not just cute, fuzzy, little tiny mammals, but also birds flying everywhere, dropping ticks that are infected on everyone. Not really, but this is how these things spread. We don't really think about that. We don't think about the bird landing in our yard, getting close to the dog, and then having a tick fall off after a meal and then jumping on the dog. These are things that have to be taken into consideration, though. And then with the vectors, as was stated earlier, you have two different ticks that are known to carry Lyme disease. You have Ixodes scapularis and Ixodes pacificus. Different parts of the country, different hosts. Some of the hosts on the East Coast that we would have assumed that the Ixodes pacificus would have enjoyed, they don't. So a lot of the most recent research is very surprising and very enlightening, and hopefully we can put that to good use. And then there's also what I know is a very controversial aspect of Borrelia burgdorferi and the various Borrelia. What kind of physical state does it take? We all know about the vegetative or the live form, the beautiful little spiral that it forms, but there are also forms that are round bodies, that are thought to have lower levels of metabolism, that have different methods of survival that can actually, once they've dealt with environmental concerns, when the environment is nicer, they pop back up and suddenly they're back in the vegetative state. But a lot of this research has been a long time coming. And so one of the things that as I was kind of brushing up on all of the Lyme disease, most recent research is the fact that there are now 24 known Borrelia species. That's astonishing. There's so many of them. And you can find multiple Borrelia in one tick. You can find multiple Borrelia in one person. And really, we concentrated very much on the Borrelia burgdorferi, but I was really excited about the fact that, in a science way and not in a good way, but exciting science way, not for people with disease, but that there's a newly discovered Borrelia mayonii that the Mayo Institute isolated, and now this is considered to be maybe not as great a cause of Lyme disease as burgdorferi, but mayonii is considered an actual agent of Lyme disease. In Europe and Asia, it's caused by other Borrelia, Afzelii and Guerinii. And then a lot of those other Borrelia, the other 20 species, can cause these tick-borne relapsing fevers and other infections. And some of them are just so rare that they're not classified yet, and we don't fully understand them. And so the whole process of getting the Borrelia into our bodies and getting disease begins when a hungry Ixodes nymph feeds on an infected host. So Borrelia will enter the tick gut, and then it sticks onto the gut. makes some changes on its surface proteins. And then when the Ixodes jumps onto its next meal, the Borrelia are in the tick saliva and they get injected into that new host. And so from there, Borrelia can enter the circulation. As Liz was saying earlier, they don't like to hang out in blood. They're very unhappy there. So they enter the circulation to go and colonize other sites. They go to the joints, they go to the hearts, they go to the bladder, they go to the brain. We've seen this in animal models, and we've seen bits and pieces of it in human patients, but there's still a lot of pushback on it for some reason, even though we know that these bacteria have a tropism for these sites. They have receptors on their little spiral bodies that recognize specific proteins, specific integrins that tell them, go to this site, go to this site. You will be happy, you will be safe, you will not be the target of an immune response, which again, the whole point of its existence is to avoid detection by various immune systems that are trying to get rid of it. And so we don't really have to go over this, but I did think this was interesting, which is also from the Bay Area Lyme Foundation website, conveniently. Speaking about the different types of lesions, I was shocked by how many different lesions are finally now being accepted as being, if not Lyme disease, then absolutely related to Borrelia type infections. Interestingly, the CDC has this thing where your rash has to be greater than 5 centimeters in order for it to be a true erythema migrans. I have no way to know what to make a judgment on that scientific basis. I do understand that the erythema migrans is actually the response of the tissue to the Borrelia there. So the tissue is responding to the presence of the organism. So the development of this and these patterns differ by region. I was reading in one publication, they said that in Maine, only 20% of patients had an erythema migrans, which is shocking. And so there's this nice paper that we just saw. So the early serology only showed 29% positivity. And the interesting thing is that when it comes to the immune response, it's very likely that antibiotics, immediate dosing of antibiotics is likely partially responsible for that. If you have antibiotics that get rid of most of the bacteria that are available to generate an immune response, the only ones that are left are hiding out somewhere, the immune system doesn't think that it has to go on and expend all this energy to make a much bigger, much stronger, much more specific response. It's already taken care of the problem, it's gone. But as we know, that might not be true. And so again, Lyme disease for a long time has been called the great pretender because all of these symptoms are so vague, so general. It's unless you can say, I had a tick, I had a bite, I had a rash, I have something, yeah, I'm in an endemic area, it has to be this. It's very difficult to say what it really is. And in the long term, if it's not treated, and even if it is treated, sometimes you can end up with very severe diseases like arthritis, carditis, neuroborreliosis. There are various spirochetal diseases that have all of these different long-term diseases as a similarity between them. And so when I started my PhD in a Lyme disease lab, and we were studying arthritis, and my boss said, what do you want to study? What do you want your project to be? At the time, I don't know if anybody remembers this, but Celebrex and Vioxx were very big. Celebrex had just gotten Pfizer $8 billion a year. It was huge. So unfortunately, it was also a reflection of the reason why our infectious disease studies and departments are in such sad shape, is because for a long time, science said, oh, we've conquered infectious disease. Look, we got rid of smallpox and we're on our way to getting rid of everything else. We don't need to spend all this time and effort on infectious disease immunology. Now we can focus on autoimmune disease, and that's where all of the immunology went for a long time. So the drugs like Celebrex and Vioxx are part of a group of drugs that are actually the most commonly used drugs in the world. You're all familiar with them. They're NSAIDs, non-steroidal anti-inflammatory drugs. These drugs have a very specific mechanism for how they work. They block enzymes that are involved in the production of different lipids, which are called eicosanoids. So leukotrienes, prostaglandins, thromboxanes, these are all made from lipids that start out in the membrane, and different enzymes work on them, metabolize them to create these different lipids that then signal through receptors, go through the body. These lipids are the reason why you can take baby aspirin if you have clotting problems. They're the reason why you can't take ibuprofen if you are prone to heart problems, or why you can't take COX-2 inhibitors if you are prone to heart problems. These NSAIDs and affecting the production of these lipids. And so NSAIDs in particular work as inhibitors of a couple of enzymes called the cyclooxygenase, or the COX enzymes, COX-1 and COX-2. And so they were the primary target of a lot of drug companies working on arthritis, rheumatoid arthritis specifically, for a very long time, and it worked quite well. But I thought it was very surprising that nobody had bothered to look at whether or not these drugs worked in infectious arthritides and what impact that had, because all of these lipids don't exist to cause autoimmune disease. They exist because they were created and evolved to do something for the immune response. So infection is what the immune response is for. So what does this family do, the cyclooxygenase products? And what does this family do, products of the lipoxygenase or the LOX enzymes, the leukotrienes? These were also targets for arthritis treatment. And so we know shockingly little about what these do to the actual immune response when it comes to infectious disease. And so to study this, most of the time, we are relegated to using mouse models. In this case, we use experimental Lyme disease, where we inoculate mice subcutaneously in the foot pad. For us, we do this because we're interested in the arthritis, not the migration of the bacteria to the joint. And so if we inject in the foot pad, we know pretty much when right around when those bacteria are going to actually get there. So that way we have a much better time frame that we can examine. So most studies in animals, in mice, are conducted in what's called the B6 mouse, the black 6 mouse. It's the most commonly used lab strain, and it's very smug. Everybody has to use the black 6 mouse, and it's very unfortunate because it's not a very good model for Lyme disease. It doesn't really get Lyme disease that badly. It's a very moderate disease. So instead, if you look at some of the early studies of Lyme disease, there's a lot of work that was done to look at 20, I believe, up to 20 different lab strains to see who was susceptible, who was resistant. And so in this case, our lab used the mouse that was called the C3H mouse. It's a susceptible mouse. It develops a severe Lyme disease, even if it gets a very small number of bacteria. And then we also use a resistant mouse, the DBA2J or DBA. It develops a very mild Lyme disease, even if you give it a whopping number of bacteria. And so we like to use both of these strains so that we can look at both, okay, what happens when you have the same inoculum, the same culture of bacteria, and you inject mice that are supposed to get very, very severe disease, what kind of signaling molecules are occurring there, and then what happens when you use those same bacteria in mice that are not supposed to get anything? Are the same systems involved? And so the first thing we looked at was the COX-1 and COX-2 enzymes. So this is what a normal response to Borrelia burgdorferi infection looks like in these mouse strains. Here you have the arthritis severity score, which is based on histological staining and the number of inflammatory cells that enter into the tissue or leave the tissue. And you can see the C3H in white have a much more severe severity score than the DBAs. They get almost no infiltration. And then here we have real-time PCR data on the number of bacterial genomes that are in the ankles of these mice at different time points. And you can see it really goes up at day 7 and then starts to go down. But we really can't detect anything at day 3, even though we injected those bacteria into the foot pad right there. They're still not at a high enough level to detect with real-time PCR, which is still pretty sensitive. And so this peak at day 7 correlates to right when the arthritis is really getting severe. And then when you look at histology here, so this is the ankle joint, this is the tendon that just kind of goes over the paw. And here we have the susceptible mice. Here we have the resistant mice. This is at day 0, everything's the same. You look at day 10, and you can see how thick this is, how many purple spots there are. They're all clumped together. You can't even tell different spots from one another. That's because there's that many inflammatory cells in that joint at that time. A bunch of these are neutrophils and macrophages, those innate immune cells that I talked about, the first responders. In the DBAs, the resistant ones, there's a little bit, but not much. And then at day 35, there's still a little bit, but it's going down. It's certainly not as severe. And the resistant mice are pretty much the same, are back to baseline. And so going back to, one of those key tests that they do for Lyme disease, looking at immunoglobulin production, you see here we have Borrelia-specific IgM. That's the first immunoglobulin that you make. It pops up at day 3 and then steadily goes down in both strains of mice. And then IgG, which, as I said before, it's a much more specific. The immune cells undergo this maturation, this crazy mutation to become more specific. And so that's why it takes a little bit more time to get this to peak. But it eventually comes up and then starts going back down because bacteria is cleared. So what happens when you don't have those cyclooxygenase enzymes working? In models of rheumatoid arthritis, you can almost completely get rid of the arthritis with these drugs in the models. Oh, sorry. So, like you said, a little bit out of practice with talking to real people again. First of all, we didn't know if these lipids were involved in Lyme arthritis. Maybe the reason why nobody was publishing on them was because somebody had looked at them and they were like, "Oh, this is negative data. This is terrible. We're not going to talk about this. Let's move on." And so we had no idea. And so we collaborated with a group at UCSD who does lipidomics. So that's the same thing as genomics and all the proteomics. You pretty much just throw everything at the wall and see what you can pick out of the spots. And so we isolated these big piles of lipids from different ankles at different time points post-infection, the C3H and the DBA, and we looked at, I believe, 135 eicosanoids. We certainly didn't find them all, but this was the first time that a lipidomic analysis had been done on an in vivo inflammatory response. Before that, all of it was done with tissue culture cells. And it's not important exactly what these lipids are. What is important is that the C3H, which have more severe disease, have much more red in many of these lipids at the peak of the inflammatory response, day 7 through 14. Red is an increase, whereas the resistant, not very much. And so we did various experiments where we treated mice with COX-2 inhibitors. In this case, we put Celebrex in their mouse chow and then examined their ankles at 35 days after infection. And so again, with normal mouse chow, the mouse ankle, you can see that it's pretty cleared up. There's not a lot of cells hanging out around here. But when the mice were given Celebrex chow, even at day 35, they still had this blazing inflammatory response, which blew our minds. That didn't make any sense. We were giving the mice this drug that everybody is taking for arthritis. And here we thought, well, mice have arthritis. But no, uh, they still maintain this high level of inflammation. Now, COX-2 inhibitors didn't make it any worse. It just made it so it stayed bad for a really long time. And when we looked at mice that didn't have the COX-2 enzyme genetically, we got the same results. The mice didn't resolve their arthritis. So then we wanted to see, well, usually COX-2 is considered the bad guy. It's the pro-inflammatory guy. So what about COX-1? COX-1 is supposed to be the good guy. It regulates the vasculature. It regulates homeostasis. But there were a few reports here and there that it was also involved in arthritis. And it could be if it regulates the vasculature. The vasculature is how immune cells get to places. And so we infected mice that didn't have the cyclooxygenase 1 enzyme, or we treated them with the COX-1 inhibitor, and it didn't really affect their arthritis, but it affected their immunoglobulin responses, which was very surprising to us. So we found that here in the white, you have the COX-1 knockout mice, and as time goes on from day 0 to day 24, they have increasing levels of IgM. If you remember when I showed you that original graph, it peaked and then it started to slope down while the IgM started to go up. But that didn't happen when these mice didn't have COX-1. And then their IgG wasn't nearly as high as the wild type mice. And so this indicates that something that's called class switching. So that's the fancy switch from IgM to IgG that requires the genetic mutation, the maturation. And this implied that by not having the COX-1 enzyme and the lipids that it makes, that these B cells weren't able to switch to the more specific, Borrelia-specific IgG. They just kept making IgM, which is not as effective as IgG at clearing the bacteria. And so various studies later, we found that we measured from serum 2 different cytokines, IL-6 and IL-17. Cytokines, same type of thing as the interferons. Interferons actually involved in this group somewhere. And here we looked at mice that were treated with a COX-1 inhibitor, because really that was a very important thought, right? I mean, we're not going to have a lot of human COX-1 knockouts running around, but we are going to have people that are taking COX-1 inhibitors. And so when we looked at their IL-6 and their IL-17, which are necessary for that immunoglobulin class switch, when the mice were given that COX-1 inhibitor, they didn't make as much IL-6, and they made almost no IL-17. And we could reverse this by giving the mice back IL-6 and giving them back IL-17. But it really kind of begged the question, if a diagnosis depends on seropositivity, but a patient is taking medication that alters antibody production, including taking antibiotics that then reduce the trigger for producing the antibodies, then where does that leave a patient who has actual Lyme disease but doesn't have the seropositivity? So we also wanted to look, as I said, at this other branch, the lipoxygenases that make leukotrienes. Leukotrienes were a very big thing for a while. They're definitely involved in arthritis, but the thought was that eventually studies found that maybe we can inhibit leukotrienes when it comes to things like allergy and asthma. That's cool. But if we try to inhibit it in something like arthritis, it turns out that they're a little bit too important for the whole immune system that's going on for us to really block them for relief of arthritis. We didn't know that at the time. So we looked at mice that didn't have this enzyme, that couldn't make leukotrienes anymore. And once again, we were surprised. Leukotrienes in rheumatoid arthritis models are involved in causing very severe arthritis. But when we gave 5 lipoxygenase knockout animals, Borrelia burgdorferi, inoculating them with Borrelia burgdorferi, on the top you have wild type ankles, bottom you have 5 lipoxygenase knockout ankles. Day 14, we expect to see a bunch of cells around, a bunch of inflammatory cells. That's what all these dark purple dots are, or the nuclei of those cells. At day 28, in a wild-type mouse, there's still a few cells, but it's starting to resolve. But here in the 5-lipoxygenase knockouts, they just have more severe disease. There's more inflammatory cells. And again, we see this curious effect on the development of antigen-specific or Borrelia-specific IgG. And we found that it turns out that the macrophages, which are two of the innate immune cells at the beginning, the first responders, it turns out that when they don't have the 5-lipoxygenase, they can't eat Borrelia as well. They're just not that good. Whether or not they're hungry or not, I don't know, but they just can't eat them very well. And that sounds like a joke, but there are actual eat me signals on cells that tell macrophages to eat them. So a macrophage may be hungry, but they can't find an eat me signal, or cells may not have the eat me signal. In this case, it looks like the macrophages weren't hungry because they didn't eat the Borrelia. And then when they needed to eat the neutrophils that had gone in and they had eaten the Borrelia and then they die, which is what they do, the macrophages are supposed to go in and clean up on that. They didn't. So everything was pretty much just hanging out and just causing a chronic inflammation. And so another way to be kind of more specific about targeting inflammatory leukotrienes is to look at the receptors for them. And so we looked at mice that didn't have the receptor for leukotriene B4, which is the really bad pro-inflammatory one in rheumatoid arthritis. And when we looked at joint inflammation in those mice, so they could make leukotrienes, they just couldn't presumably signal with them, with this specific one. At day 14, it's about the same arthritis severity, but at day 35, when the wild-type mice were resolving, the BLT knockout mice were actually getting more severe. And you can see again here that there's actually a change in the joint architecture because there's so many inflammatory cells still at this time point. So since the 5-lipoxygenase was involved in cell death and eating the bacteria or eating the immune cells, the dying immune cells, we wanted to see if we were changing the number of cells that were going in and what they were doing. So at all time points that we looked at, the mice that couldn't signal through BLT1 had more neutrophils, so more pro-inflammatory cells going into the joint, but they weren't even dying. So they weren't undergoing normal cell death. They were going in, eating some bacteria, and then they were undergoing necrosis. And so that was triggering a chronic inflammatory response. And so when I moved to do my postdoc and my fellowship, I couldn't stay in the icosanoid field. Like anybody who's in science knows, you have to kind of change it up because otherwise they say you're not taking big enough risks, you're not learning enough new things. So when I moved, I changed to a new lipid, a new group of lipids called the sphingolipids. And they're also created from membrane lipids through multiple enzymatic responses to make my favorite lipid, sphingosine 1-phosphate, or S1P. This gets secreted out of the cell, and then it binds to albumin or HDL. Albumin is actually the most common protein in circulation versus IgG, which is the second most common. So intriguingly, both HDL and S1P have been correlated with susceptibility to infectious disease, and actually S1P has received a lot of attention recently for being a marker of severity of COVID and likelihood to progress to severe COVID and chronic long-haul COVID. So, pretty excited about that. And conveniently, there are now 4 FDA-approved drugs that target the receptors for S1P, and there are 5 S1P receptors. They are on different cells. They tell the cells to do different things. But 2 of the most important things that S1P receptors do is tell immune cells where to go and what to do when they get there. And they tell the vasculature whether or not to be leaky or whether or not to be activated. So I'm pretty excited now that I have my own lab about the possibility that these lipids could be involved in the in whether or not Lyme disease is cleared, resolved, whether or not it becomes chronic, and how differentiating whether or not a patient is possibly going to progress to a chronic state even if they're treated with antibiotics. So, and very funny is the fact that this is actually from the last figure of my dissertation where I was studying completely different lipids. And I looked at those BLT1 knockout mice, and the gene that was the most downregulated was the gene for the protein that makes S1P. And those are the mice that don't resolve, that their cells don't undergo apoptosis. And so, I don't know, I guess I was just destined to have these two worlds collide at some point. So, right there. So, through all these studies, what have we learned? Well, we realized that we absolutely understand autoimmune disease, frighteningly, much better than we understand infectious disease, and that's because it's been neglected for so long. And we also found that treating symptoms without any thought to why those symptoms are there can have some unintended consequences. And again, we need to look outside the box. Cytokines are not the end-all be-all. They're very easy to isolate, they're very easy to make, and they're very easy to give. But that's not necessarily the reason to study something. We need access to more patient samples, so excited about that. And we need physicians to acknowledge that infectious diseases have a lot of variability because you're not just dealing with one organism, you are dealing with at least two organisms. You're dealing with the host and you're dealing with the other organism that's trying to stay alive inside of that host. And so until people will realize that, we're going to have to do a lot more experiments to kind of change people's minds. And so I just have to acknowledge the people at University of Missouri where I did most of this work. Charlie Brown was my PhD advisor. That's his real name. SBP, my cohort here who have helped out, and then help from other institutions. So, thank you. I went over.
That was amazing.
Oh, God.
Okay, thank you for your patience and hanging in there. I would like to introduce Dr. David Haney. David Haney is a biochemist who helped develop the first test for hemoglobin A1C that was used at Harvard to correlate diabetes control. He served as an NIH grant reviewer and journal publication reviewer. He's the president of Haney Associations in La Jolla since 1993. He received his bachelor's from Boston University, his master's of science and PhD from Northwestern. And more importantly, he's raised two sons who went into computational science and is here to talk about his son, Sean Haney, PhD, whose last publication was out of Scripps Institution of Oceanography at the University of California at San Diego. Thank you, Dr. Haney.
Thank you. My presentation may seem dry and clinical. Sean was a very special person in so many ways. But in order for me to complete this presentation and hopefully provide some insight, I need to be very clinical. And I guess I have to wait for something to happen before I can move on. Maybe we could just leave it like this. Okay, that seems to work. A clinical piece of data that is ignored by most physicians, at least for Sean, was that he had a huge compassion for outdoor activities like mountain biking, hiking, climbing, camping, surfing, etc., where the ticks are. My son, Dr. Sean Haney, died January 1st of this year. He was 33 years old. He did not die of COVID, which he worried about. He did not die of a mountain climbing accident or a mountain biking accident, which I worried about because he's such an outdoor guy. But what he did die of is of cardiac complications due to tick-borne or zoonotic infections, like Lyme disease. This is despite the fact that Sean had at least a half a dozen cardiologists examine him over 10 years. Unfortunately, we all do not worry enough about zoonotic infections, and not enough is known about Lyme and co-infections, especially cardiac complications. Sean was the most socially adjusted genius that I know. He was brilliant and at the same time, the social organizer. Sean was a unique position in which he was a postdoc at Scripps Institution of Oceanography and also a principal investigator. My speculation is that Sean was infected due to a tick exposure in March 2009. Sean and his brother both knew that they had significant tick exposure from a backpack camping trip to Big Sur. Even though they did not have the classic bullseye rash or any rash, I recommended that they get antibiotics as a precaution. It's interesting that both Sean and his brother had severe tonsillitis about 3 months after the Big Sur event. Sean's treatment took several drug attempts, starting with doxycycline, while his brother was successfully treated with azithromycin, which can be effective against Bartonella. I think that for many people that have chronic persistent zoonotic and tick-borne infections, there are multiple exposures. Sean also had significant exposure to what's called cat scratch disease, which is caused by Bartonella, and I think somebody pointed out is unfortunately treated as a minor disease, and it's not, because his girlfriend had a cat that scratched him frequently. He also had exposure to ticks while mountain biking in Colorado. Another probable exposure is this one to Bartonella, which occurred on an airplane flight. Immediately after he got off the flight, he got to his hotel room and sent me this photograph, which is possibly a bite from a flea, but it's a classic Bartonella streak rash, which we didn't recognize at the time. Zoonotic infections with Borrelia, Bartonella, and Babesia, or Lyme disease, can lead to a multitude of unusual symptoms. In retrospect, Sean's unusual medical issues that may be related to tick-borne infections include the severe tonsillitis, which repeated several times, including 3 to 4 times in 2017 to 18 alone. Infection of lymph tissues, including the tonsils, is a symptom of Bartonella. Sean had various cardiac symptoms, including syncope, chest pains, breathing difficulty, arm pains, resulting in several cardiac echoes. Cardiologists could not figure out the problem, despite several EKGs showing inverted T waves and conduction delay. Inverted T waves and conduction delay are known to occur as a result of Borrelia and Bartonella. I have since learned that Columbia's Lyme group will put someone with a conduction delay on a continuous monitor. Combine this with the most recent echocardiogram, which shows mitral valve dysfunction, then it appears pretty obvious that Sean suffered from some cardiac problems that no one wanted to own. Sean had Raynaud syndrome followed by progressive general weakness, which resulted in the ALS label. Sean had severe anxiety that even resulted in a trip to the ER, possibly caused by cardiac issues, although they'd never found anything. Sean had minor difficulties controlling bladder function, which is unusual for a 33-year-old, but it's a known symptom of Lyme disease and also a symptom of dysautonomia. Sean had the classic mobile pains or prickly feeling as described by Horowitz, moving from arms to hands to chest to legs to feet. Sean had the insect bite that I just showed that had the classic Bartonella streak. He had erectile dysfunction, which is also a dysautonomia symptom, but had tests showing low testosterone, low TSH, and low growth hormone. Sean had sudden hearing loss, which really surprised him, and he wanted to have it treated right away. And none of the physicians knew what was causing it, but it's a rare but reported symptom of Lyme disease as well as dysautonomia. Sean had other classic Lyme symptoms, including headaches, nausea, brain fog, stiff neck, severe fatigue, and oddly enough, gingivitis, not something you normally see in a 33-year-old. But interestingly, vets use gingivitis in cats to follow whether or not the cat is infected with Bartonella. After failing to get anywhere with traditional healthcare physicians and being told that he was not smart enough to study his own disease, which they couldn't figure out, Sean saw 3 different Lyme specialists. His test results varied a little bit over 3 years, but he was consistently positive for an unknown species of Borrelia, which they had tested Miyamotoi, Hermsii, and Terracate. But in addition to the positive test for Borrelia, he had clearly positive tests for Babesia and 3 different species of Bartonella. Most local physicians are under the mistaken impression that there is no Lyme disease in California, but people travel, and tick-borne infection is more than just Borrelia burgdorferi. In fact, the CDC's map of TBRF Borrelia, as well as academic studies of Babesia duncani, show that these zoonotic organisms are more prevalent in the West than the East. Sean lived near Seattle, San Diego, Boulder, and visited Big Sur, certainly places that he could be infected with TBRF. A study by UC Davis showed that nearly 20% of the ticks in Santa Clara County tested positive for Bartonella. It seems that some physician education is essential. Sean was treated with a variety of antibiotics and IVIG. He seemed to be slowly getting better, though not regaining his muscle strength. Then he was started on rituximab, which he had because he had some autoimmune antibodies. This was probably a huge mistake. After that, his heart problems became more obvious, resulting in yet another echo. Unfortunately, no one recognized the potential value of a troponin T test, and the cardiac issue went undiagnosed. However, Sean did get tested for CK isoenzymes, which showed an elevated CK-MB, which is a fairly specific test to show myocardial damage. He also had elevated CK macro type 1, which is commonly elevated due to an autoimmune problem, probably caused by Borrelia and Bartonella. Sean probably died of a type of tachycardia known as torsade de pointe, or polymorphic ventricular tachycardia, or some other ventricular arrhythmia, which can result in sudden death if not treated immediately. A person can be shocked out of torsade de pointe. Unfortunately, I believe that many physicians do not know how to interpret some medical test results like the CK isoenzyme test. So what have we learned? The traditional healthcare system makes it very difficult to get diagnosed and treated for zoonotic diseases. They improperly believe that no one in California can have tick-borne or zoonotic infections. They reject valid testing, even though there are no other, that no other lab can do certain tests that they do. They do not recognize the symptoms, and they even miss symptoms of heart disease caused by tick-borne infections. Some tests can be very helpful, but many physicians do not know how to interpret them, like the CK isoenzymes. Unfortunately, once the healthcare system applies a label like ALS, MS, or Parkinson's, which there are no definitive diagnostic tests for, that label sticks in your record and they stop looking for the real cause. Again, it seems like physician education is critical. Number two, people should be on the proactive lookout for the smallest cardiac issues and pursue them. I believe that most Lyme docs need extra training on potential cardiac problems caused by tick-borne infection, as the normal healthcare system will simply write these small deviations off to a variation of normal. Number three, as Horowitz recommends, do not use extremely powerful immune suppressants like prednisone or rituximab until the infection is gone. Zoonotic infections can cause elevated autoimmune antibodies like GAD65. These elevations should not be taken as proof that a strong immune suppression should be used. Number four, do not expect to get better quickly, as you would from most other infections. It's more like tuberculosis, which can take years and many protocols to treat. Be wary of falling into treatment for something other than the tick-borne infection. Because of the time it takes persistent Lyme to respond, some, like Sean, may question the Lyme diagnosis and fall back on traditional healthcare system treatments, which can be harmful. Number five, Bartonella can be a more dangerous zoonotic organism than Borrelia. It seems to me that the literature is pretty clear that the only current clinical treatment to eradicate Bartonella is with a minimum of a 2-week course of an aminoglycoside like gentamicin. Unfortunately, none of the Lyme docs were very excited or willing to use gentamicin. As a result of this, and because of the potential for hearing damage, Sean ruled it out. Finally, although DEET is promoted for mosquitoes, it's effective for ticks and fleas. Please be safe.
Thank you, Dr. Haney, for sharing your son's story with us. We're going to move on to a question and answer period. Just in case anyone leaves, I want to throw out 2 things. I want to thank our sponsors, Quidel, for helping us bring this together tonight and for coming and attending this event. And I also want to point out that I didn't say this earlier, but the Bay Area Lyme Foundation is supported by a family grant, a foundation grant. So all the donations that come in go 100% go out to research. None of it goes to overhead or to us. And that's a pretty special thing about our foundation. So I'd like to invite our panelists, our speakers, to come and take a seat up here. I know that there's at least one question over there. So Liz, is there a microphone for you? Just pass it around. All right, so the first question, I know, okay, no. Are there any questions? Yes.
Sorry.
My question is very much a technical one, and it relates pollution from the what the trigger for that is biochemical.
So, I'll just take my mask off. The question was, what's known about what, I guess, essentially what metabolic triggers or what biochemical triggers are there for Borrelia to revert back to some type of vegetative. Most recent that I saw, which oddly enough had pictures that were very, had a lot of the same information that the earlier one did, which is just that it has something to do with that was they had looked at that for whatever reason, if there was a specific amino acid deficit, and then when it was added back into, they could revert back to viral replicating spirochetes.
A recent kind of repeat of that work, it sounded like,
Yeah, it was relatively recent. It
Okay.
definitely wasn't, but it's the same, like, look, we probably in a deficient environment, they do this.
Yeah, okay.
Previously, people had said, well, it's just because you're not treating the cultures right, so it's an artifact.
Variability in positive testing. Geographic location. That pick it up as well in some regions versus others. Timing and geography.
Yeah, so I think timing, so the first question is when you're doing serology, will the timing of when you actually take the test relative to the beginning? Very likely it does, and a lot of these are early disease. So we do the best we can to ask people what they remember. You know, do you remember a tick bite? Was it attached? How long have you felt sick? And most of our cohort in that early cohort is probably, it's all less than roughly 21 days, but the ones I was showing were a little bit earlier. So it's very possible that the immune response hasn't had a chance to develop. The part that, and I have a collaborator right now, and he calls it curious, is when we look at the second draw and we look 2 to 3 months out, and Dr. Blaho did a really nice job talking about class switching, which, and affinity maturation, which I didn't want to bore you with, you should see the IgG come up because that's the hallmark of Lyme disease and that's what's on the CDC website. And that's not what we see in our data. But what's kind of cool about our data is we see this at different sites and different geographic locations and over multiple years. So I don't think it's an artifact. I think we need to understand more what's actually happening. And I also, it's really interesting when we started this project, we had like these really lofty goals that we were going to culture the Borrelia from people's blood in early disease and get all the different isolates and strains and species and be able, and you know, we got 2 isolates out of culturing. This poor graduate student, he just looked in a microscope all day long looking for stuff to grow and it just didn't. I think that's one of the real hard things about Borrelia. It does, you know, it's very difficult to culture. It doesn't hang out in the blood. And we don't have a good direct detection test. I mean, really, that's what we need to be able to say, yes, you're infected with Borrelia and we can detect it. So think about, not to diverge to COVID, but I think one of the, if I could call it a silver lining about COVID, is it actually is people talking about the importance of accuracy in diagnostic tests and why it's important to actually to detect the virus. And before we had vaccines, to detect the antibody, to know if you'd been infected, and really trying to show the importance of having accurate diagnostic tests and all of the challenges that we saw last year. These are all challenges in Lyme disease. So, and there could be a lot of influences. And there's also the whole host issue. People are different, and and I think it might just be time to reevaluate how we're looking at this. And I want to do this is a very long answer to a kind of a simple question. But one of the things that's actually going on right now is LymeX, and it's a public-private partnership between the Department of Health and Human Services and Stephen and Alexandra Cohen Foundation. And one of the pieces of LymeX is a diagnostic moonshot prize. And it's exciting because we're going to be harnessing some of the resources the government has, that a private foundation has, and really trying to get better diagnostics, especially for direct detection. So there'll be more information about that later this year.
Oh, I'm sorry. Thank you.
Yeah.
Having watched son go through a multitude of tests for a variety of these organisms. Back to your question about, does it depend on when? Does it depend on where the test is done? Yes to both, in our experience. And the reason that it depends on when is because it appears that the Borrelia and even to some extent the Bartonella go into hiding. And when they are in biofilm, your immune system isn't reacting to them. And so the level of IgG that you would have, I'm talking specifically for, chronic type Lyme, not the immediate type Lyme, because they didn't need when they got infected. But these IgGs will go up and down during the during the disease, depending on what you're taking. If you take a lot of antibiotics, they will actually go up. And then if you stop taking antibiotics, they go back down, but they don't go away.
I'm not sure if it was specific to Lyme in general. Rethink this model that I was taught a long time ago at medical school about IgG response and the convalescent, IgM immediate and IgG convalescent. Because at least in Lyme and possibly in general, it doesn't always go that way. And I'm wondering if comments about that, either that are Lyme specific or in general, could it be this migrating in and out of different dormant forms and forms that the immune system starts to then signal again? And you can have persistent IgM 5-10 years into chronic Lyme.
Oh yeah, I mean it's part of the problem is you're trying to find. I mean it's a screw needle, but it is very much a needle that it's so difficult to find these things, even if you're not taking from human tissue and you just have a tiny punch, even when you have a mouse, like an entire mouse heart, you can section the whole thing. It's almost impossible to find the things. once it reaches a certain stage. So, and I feel very confident that it probably just like a lot, I mean, TB, sits there and makes its own little case for itself. It's just easier to know that it's there, right? Because you can see the the granuloma. Lots of infectious organisms do this, and so that's why you get these nice, dips and then peaks of the immune response, because it takes a lot of energy to make, especially to make clonal B cells or to keep making, and if there's no need for it, shut that off. But then that's probably when the Borrelia decides, oh, well, because I mean, our metabolism changes when we have a raging infection, right? Like, for instance, my mom just had sepsis a few weeks ago and acute kidney failure. Her glucose was over 400, and they kept pumping insulin into her, and they couldn't get it to drop because that's just the response, that you have, right, to that kind of an infection. And so I'm sure the Borrelia and other bacteria can sense that. They sense whether it's a sugar or an amino acid or a specific peptide, and once that situation calms down and the immune system goes, oh, it's clear, coast is clear, we're good, we did it, guys, high five. And the Borrelia is like, all right, now we can go out again. And maybe we need to find a new area. Maybe we need to.
I would just also add that the antibody-based tests are based on Borrelia burgdorferi, and we have other strains and species, and so the antibody testing also has that challenge as well. Other questions? Yes?
If I led the way there, so can I ask a question of Dr. Haney? Yeah. So Dave, clearly one of the issues with tick-borne disease generally is that the medical community is not very well geared to recognize, to accept, to treat them. So I'm just wondering if, from your experience, kind of looking forward, what are the things that come to mind that we might do to help better educate the medical community?
First of all, I think that they need to be laboratories like Hygienics and Galaxy, which do a lot of the tick-borne analysis, provide correct results. And our scripts, our experiences right here in San Diego, those places would accept those tests. They think they're bogus. And how can you say something is bogus when they're an approved clinical testing laboratory? I just didn't understand that. So I don't know if that comes in regulations where the government says if you're going to provide healthcare services, you must accept these tests from approved laboratories. I don't know, but it just very frustrating.
What was their reasoning for that? For calling them, for saying that they're bogus?
They didn't really give a reasoning. They just said that we've seen this before, bogus. And it's not just Borrelia burgdorferi. The same thing went for Bartonella, and I don't understand how you can argue with a fish test finding DNA. I wouldn't accept that test even though they found the DNA.
Back there?
That's a really good idea, the peptide mapping. So,
But you can then challenge
yeah,
and try to amplify the process. You know, as
start maybe.
Well, I mean, mouse models, of course, have their limitations. I think.
You could do germline analysis.
Oh yeah.
So it's.
Oh yeah. No, I was going to say that one of the problems is that in the field using mouse models, a lot of times nobody goes. I mean, I was kind of given a hard time for going to day 35 after infection instead of just stopping at like day 21 because everybody in the field said, oh well, it's on the way out. It's fine. Don't have to look that far. And there were times when I took the mice out to like day 60, and a fun surprise that we got is that the DBA mice, which aren't supposed to get anything, suddenly started getting something. Yeah, that was concerning. At first we thought it was a fluke, but then it kept happening if you just let them go long enough, even though when you go to do PCR, real-time PCR we would take the bladders out too and do PCR or try and culture them. We would take ankle joints out, PCR, culture, hearts, same thing. No PCR, nothing would grow out. But if you leave the mice long enough, they'll start to develop arthritis. And they'll get arthritis just as bad as the susceptible strain. So, and kind of the same thing with the antibiotics. We could treat mice with antibiotics, and they would resolve. Nothing would, actually that was a little bit different, nothing would grow out, but when we did PCR for forever out, we could still keep finding little bits and pieces of Borrelia DNA. So, yeah.
Not find any patterns.
Could be.
So
And since they're different genotype, different backgrounds, that's the nice thing, I guess, about having the two different backgrounds that have been mapped. You can say, well, this guy has all these mutations, and this. Is there a question over here? Yeah.
How do you, if you do have a diagnosis, which our son has been diagnosed, how do you know that you're getting after actually, like you said, is there a resource bank out there for like doctors who are very knowledgeable, who have had a specific kind of training, something like that? Because it seems.
So there's the International Lyme and Associated Diseases Society, ILADS, ILADS, and they are a physician. Medical practitioners who consider their training Lyme literate, which is hard. There's no official certification course and whatnot, but they do have training courses that they offer as an association. Will provide stipends for physicians to go do rotations with other physicians who are trained in tick-borne disease and train that way. They provide CME conferences. Also have a physician referral directory for physicians. Not easy. I wish it was a perfect.
Is that a helpful thing? Contact tracing, like for instance, our situation is because but the camp didn't necessarily notify us. Lyme disease, you should have probably one of the appropriate tests. That's something that researchers.
No, they should be. They should be there. So positive cases should be reported to the state departments of public health. But it is rife with problems in reporting that data. Um, I wish I could say yes, but
Yeah, and I think that so there's a couple of things. I mean, we really haven't talked at all about prevention, but prevention is really, really important in Lyme disease, to be aware of ticks, to do tick checks when you're out, when you're in the woods. Lyme is done. Again, this is a little more pre-COVID because they're educating kids at summer camps, outdoor schools, and educating park rangers, because I think if you can start those good habits early on, that can help people be aware of ticks, and prevention is really important. And also knowing areas where there are a lot of ticks and there are different, but again, this is all at the state level, and public health is underfunded, and vector-borne diseases are underfunded. And but I know, like in New York State, they do a pretty good job of indicating places where there are really high levels of ticks. So I think we just need to come at it at multiple angles. And to Leah's point about the public health, to the reporting of cases. So I show a map from 2017 with Lyme cases, because each dot represents a reported case. But to be a CDC reported case, the bar is so high. So all of people with EMs that get doxy, they're not reported cases. So that's why they estimate that there are so many more people with Lyme disease. And this is a really fun fact. So Massachusetts, the state of Massachusetts has decided that the bar is too high to report cases of Lyme. So if you go on the CDC website, you can look over time and see the state of Massachusetts is completely white and there are no blue dots. And that's not because they've eradicated Lyme disease in Massachusetts. It's just the bar for reporting is too high. So, yeah, that's where prevention really comes in. And unfortunately, I wish the summer camp would have been more upfront with parents.
I'm going to speak in Tulare for the for the California Association, Outdoor Educators Association, the California Park Rangers, and I speak to. And our goal is to make sure that the people who are in charge understand tick bite prevention, what to do if someone does receive a tick bite, how to follow protocols to ensure safety. And also how to teach the children to identify and ticks. So it's something that we feel really strongly about, and we agree with you. Okay. Well, thank you all so much for hanging in there with us, and for to our speakers for sharing all of their hard-earned and hard-learned information. We'll be outside in the lobby if you have more questions, and please come and eat all that food and take some home for your families.